Humans make mistakes. Sometimes we do not have full command over techniques. Sometimes we misuse equipments. So, human error, poor laboratory techniques, and misuse of equipments cause the majority of laboratory injuries and work-related infections. The good microbiological techniques are the care and hygiene practices through which we can remain safe from injuries and infections in the lab. The first thing from which we start our work in the lab is specimen. A specimen is a sample of something or small amount of something that is taken for examination or testing, for example, urine or blood specimen. The improper collection, transport and handling of specimens in the laboratory carry a risk of infection to the person involved. Now, the specimen container may be of glass or preferably plastic. The specimen container should not leak when the cap or stopper is correctly applied. Similarly, no material should remain on the outside of the container. Container should be correctly labeled to facilitate identification. The specimen request or specification form should not be wrapped around the container but placed in separate, preferably in waterproof envelopes. Now, to transport the specimens within the facility or to move the specimens from one place to another in the lab, we should use secondary containers to avoid accidental leakage or spillage. The specimen containers should be fitted with racks to remain upright in the secondary containers. These secondary containers may be of metal or plastic. However, they should be autoclavable or resistant to the action of chemical disinfectant and should be regularly decontaminated. Laboratory should designate a particular room or area for the receipt of the specimens. Personnel who receive and unpack the specimens should be aware of the potential health hazards involved and should be trained to adopt standard precautions. The primary specimen containers should be opened in a biological safety cabinet where disinfectants should be available. Whenever we are going to use pipettes in the lab, we must use a pipetting aid. Pipetting by mouth must be prohibited. All pipettes should have cotton plugs to reduce contamination of pipetting devices. Infectious material should not be mixed by alternate suction and expulsion through a pipette. Remember that do not forcibly expel the liquids from a pipette. It is preferable to use digital pipettes if available, as they do not require expulsion of the last drop. Contaminated pipettes should be completely submerged in a suitable disinfectant and should be left there for appropriate length of time before disposal. A discard container for pipettes should be placed within the biological safety cabinet, not outside it. To avoid dispersion of infectious materials drafted from a pipette, an absorbent material should be placed on the working surface, which should be disposed of as infectious waste after use. The microbiological transfer loops may disperse infectious material. Now, if we are using the transfer loops, we should avoid the dispersal of infectious materials. In this regard, the structure of the transfer loop is important. Microbiological transfer loops should have a diameter of 2 to 3 millimeters and be completely closed. The shank should not be more than 6 centimeters in length to minimize vibration. To sterilize transfer loop, closed electric micro incinerator should be used instead of an open flame. Disposable transfer loops are preferable, which do not need re-sterilization. Similarly, if the remaining sputum samples left open, they create aerosols during drying and disperse infectious materials. Therefore, the discarded specimens and the discarded culture should be placed first in leak-proof containers and secured tops. Then they should be autoclaved, and after autoclave, those discarded materials should be disposed of into waste containers. The most important thing in avoiding the dispersal of infectious materials is the decontamination of the working area with the suitable disinfectant at the end of each work period. Each equipment is provided with the operation manual by the manufacturer. Before going to start use of equipments, we should first read the provided manual. Therefore, written protocols or safety or operation manual should be issued to the lab staff regarding the use of biological safety cabinets. If you are working in the biological safety cabinet, we must not open the glass viewing panel. We should keep the minimum materials in the cabinet. We should use sterile disposable transfer loops, as the Bunsen burner or electric micro incinerator is not permissible in the biological safety cabinet. The operator should not disturb the airflow by repeated removal and reintroduction of his or her arms. We should disinfect the surface of the biological safety cabinet after work and at the end of the day. Placement of the paperwork inside the biological safety cabinet is not allowed. Now, how we can avoid ingestion of infectious materials and how we can avoid the contact of infectious materials with skin and eyes. Remember that large particles and droplets released during microbiological manipulations settle rapidly on bench surfaces and on the hands of the operator. Therefore, disposable gloves should be worn. Laboratory workers should avoid touching their mouth, eyes, and face. Food and drink must not be consumed or stored in the laboratory. Generally, this is our habit that when we are busy in some work or waiting for some results, we put pen or pencil in mouth. This is not allowed in lab. So, no article should be placed in the mouth. Similarly, cosmetics should not be applied in the laboratory. Proper personal protective equipment should be used to avoid ingestion of the infectious materials and to avoid the contact of infectious material with skin and eyes. Like the face, eyes and mouth should be shielded or protected during any operation that may result in the splashing of potentially infectious materials. Needle sticks or broken glass can injure lab workers. Such accidental injuries may inject infectious materials into lab personnel. However, injection of the infectious materials can be avoided through careful practices and procedures, and if possible, the glassware should be replaced with plasticware. One of the very frequent and routine work in lab is serum separation. During the separation of serum, splashes and aerosol formation should be avoided. gloves and eye protection should be worn. Remember that blood and serum should be pipetted carefully and not poured. Pipetting by mouth must be forbidden. After use, the pipettes should be properly disinfected. The disinfected specimen tubes should be placed in suitable leak-proof container for autoclaving or incineration. Freeze drying is known as lyophilization, which is a low temperature dehydration process. Now, those ampoules which contain lyophilized infectious materials should be carefully opened because sudden inrush of air may disperse some of the materials into the atmosphere. Therefore, such ampoules should always be opened in biological safety cabinet. First, decontaminate the outer surface of the ampoule, then hold the ampoule in alcohol-soaked cotton to protect hands. Remove the top gently and treat as contaminated material. Add liquid to the ampoule very slowly for resuspension to avoid frothing. As you know very well the process of disinfection and sterilization, here are few terms which we are used in disinfection and sterilization. Let's define one by one. To what we call antimicrobial. So, antimicrobial is an agent that kills microorganisms or suppresses their growth and multiplication. What is antiseptic? Antiseptic is a substance that inhibits the growth and development of microorganisms without necessarily killing them. Remember that antiseptics are usually applied to body surfaces. What is biocide? Biocide is a general term for any agent that kills organisms. To what we call chemical germicide. A chemical or mixture of chemicals used to kill microorganisms is called chemical germicide. Now, any process which removes or kill microorganism is called decontamination. The term decontamination is also used for removing or neutralizing hazardous chemicals and radioactive materials. What is disinfectant? Disinfectant is a chemical or mixture of chemicals used to kill microorganisms but not necessarily spores. Remember that disinfectants are usually applied to inanimate surfaces or objects. So, disinfection can be defined as physical or chemical means of killing of microorganisms but not necessarily spores. There is another term which is often used in place of biocide, chemical germicide or antimicrobial, that is microbicide. So, microbicide is a chemical or mixture of chemicals that kills microorganisms. Now, if microbicide also kills spores, then it is called sporocide. Finally, a process that kills or removes all classes of microorganisms and spores is called sterilization. Now, there are a number of chemicals used as disinfectants. Here, we would discuss about only two chemical germicides which are the most widely and frequently used disinfectants in laboratories. The first one is chlorine. Chlorine is a fast-acting oxidant, widely available broad-spectrum chemical germicide. Chlorine is normally sold as bleach, an aqueous solution of sodium hypochlorite, which can be diluted with water to provide various concentrations of the available chlorine. Chlorine, especially as bleach, is highly alkaline and can be corrosive to metal. Storage of stock or working solutions of bleach in open containers, particularly at high temperature, releases chlorine gas, thus weakening their germicidal potential. Remember that chlorine gas is highly toxic. Chlorine can react with water in the mucosa of the lungs to form HCl, an irritant that can be lethal. The damage by the chlorine gas can be prevented by the activated charcoal commonly found in the gas mask. Alcohol as chemical germicides. Ethyl alcohol and isopropyl alcohol have similar disinfectant properties. Ethanol kills organisms by denaturing their proteins and dissolving their lipids. Ethanol is effective against most bacteria, fungi, and many viruses, but is ineffective against bacterial spores. The concentration of ethanol, approximately 70%, is highly effective. Sometimes we make mixture of alcohol and other agents like formaldehyde and chlorine to have more effective chemical germicide. Alcohols can be used for disinfecting working area, biological safety cabinets, and hands. Remember that alcohols are volatile and flammable and therefore must not be used near open flames. Principles of laboratory biosecurity. The term biosecurity has multiple definitions. In the animal industry, the term biosecurity relates to the protection of an animal colony from microbial contamination. In some countries, the term biosecurity is used in place of the term biosafety. We define biosecurity as the protection of microbial agents from loss, theft, diversion, or intentional misuse. So, biosafety and biosecurity are related but not identical concepts. Biosafety programs reduce or eliminate exposure of individuals and the environment to potentially hazardous biological agents. Biosafety is achieved by implementing various degrees of lab control and containment through laboratory design and access restriction, personnel expertise and training, use of containment equipment, and safe methods of managing infectious materials in a lab setting. Now, biosecurity refers to prevent loss, theft, or misuse of microorganisms, biological materials, and research-related informations. Biosecurity is accomplished by limiting access to facilities, research materials, and informations. Biosafety and biosecurity measures are usually complementary, and both the terms share common components. The common components in biosafety and biosecurity programs are: both are based upon risk assessment and management methodology. Both are based upon the personnel expertise and responsibilities. Both the programs have control and accountability for research materials. Both the biosafety and biosecurity programs follow standard rules regarding material transfer documentation, Similarly, training is the important common component. Both the programs establish emergency planning and program management. Concluding the principle and the principles of biosafety and biosecurity as biosafety looks at appropriate lab procedures and practices necessary to prevent exposures and occupationally acquired infections, while biosecurity addresses procedures and practices to ensure that biological materials and relevant sensitive information remain secure.